All right, let's look at that first stage presentation of the cellular adaptive immune response. If you recall from the last section we did, I presented the idea of a MHC, a major histocompatibility complex. Remember, these were used in the screening process. That's because MHCs are identifying proteins. There are two different types. These are summarized in table 12.3, but I've also put the information here. You have MHC1, major histocompatibility 1 proteins. These are found in every one of your cells, except blood cells. As I put there, it's an ID protein. This is like your security badge. If you don't belong here, then you're not given a security clearance to be in the host cell. So the MHC1 are what identify you as the host cells. Contrast that with MHC2. Those are found only in APCs. If you recall from figure 12.2, the first part of presentation, for T cells you have to have an antigen presentation cell, an APC. So APCs are leukocytes. Only leukocytes will have this role. Now, while all leukocytes can have these MHC2, the ones that are most active in the immune response are the dendritic cells. So that's why I put a star next to it. But also pretty prominent in the immune response are also your macrophages. Remember those monocytes after they mature, and B cells. So, two different types of MHCs. One found in all of your body cells as a badge, the other just found on these antigen presenting complexes. Here's how they play a role in presentation. Antigens can come in two flavors. If you remember, step 2 of the infection process is invasion. And in the handout that I gave you, there are two different ways that pathogens can exist in your cells. One of those ways it can exist is in an intracellular life cycle. These are things like viruses. We've seen some bacteria that go intracellular, as well as some protists. Remember, toxo actually will go intracellular. Or even your tumor cells, they begin to look different to your body on the inside because there's something different about what's going on genetically. Or in invasion, you can go and you can live extracellular. That means on the surface of the cells. There are bacteria that will live on the surface of your cells. Not viruses. Remember, they have to get inside your cell before they get good. A lot of fungi will live on the surface of your cells, as well as those helminths, those tapeworms, and those roundworms that we saw in the digestive tract. They live extracellularly. Depending on the lifestyle of your pathogen will determine how it is taken care of in presentation. So, phase 1 of the cellular adaptive immune response is presentation. So, if your pathogen is intracellular, we can look to figure, I don't have these numbers memorized, but if you look at figure 12.9, you can see how a pathogen will be presented if it has an intracellular lifestyle. So, typically this is occurring within some kind of phagocytic cell. Since the pathogen is inside the cell, we don't have to bring it in. It's already done whatever it's done in order to get into your cell. However, you have some vesicles called proteasomes. And proteasomes contain an enzyme called protease. And proteasomes with the enzyme protease will hydrolyze proteins. Things like that capsid that is on the virus. Remember, a virus is made up of a genetic portion, made of DNA or RNA, and then that capsid, which is made up of a protein. After we break down that capsid, we now have these different fragments of protein. Those fragments are sent to the ER. Now that's not the emergency room. So if you remember back to your intro bio for eukaryotic cells, the ER is that endoplasmic reticulum. You have a smooth endoplasmic reticulum and you have a rough endoplasmic reticulum. We're not really worried about which one of those, but we do take it over there. As it's in the ER, it then gets repackaged with an MHC1 protein. So now I have a combination MHC protein with a little bit of a fragment. So we have an MHC now with a fragment. So in the picture, they show the fragment as the blue or purple triangle, and the two orange things coming off of it are your MHC1 antigen complex. They will now form a vesicle. So you see the bubble and with the MHC1 antigen, and that vesicle will migrate to the plasma membrane. Ooh. The vesicle migrates and will merge with the plasma membrane. So PM is my acronym for plasma membrane. And then you can see there, this is where it can actually meet up with and be presented to. This is where we get presentation to a T cytotoxic cell. So MHC1 protein complexes with their antigen get presented to T cytotoxic cells. So that is in the intracellular lifestyle of your pathogen. However, if you go the other way, if you are an extracellular living thing, the first thing we're going to have to do is phagocytosis in order to bring you in. So if we look at figure 1210, so the extracellular pathway. I think I said that was figure 1210. First step there is going to be phagocytosis of the pathogen. If it's on the outside, we need to get it inside so that we can break it down. So you can see then in the next steps, and you're going to notice this is going to be very similar to what we saw in figure 10.9 when we were studying the different steps of the infection process. When we bring something in, this creates a phagosome with the pathogen. That phagosome then merges with a lysosome to create a phagolysosome. And then the lysosome with lysozyme, the enzyme within it, will hydrolyze the pathogen. And it will create fragments. So it's a pretty similar process as it was in the last one. However, this time we're not going to migrate over to the ER. And instead of forming a complex with a MHC1 in the extracellular pathway, the fragment makes a complex with MHC2. This MHC antigen complex will do what it did in the other cell. This will then migrate to the plasma membrane. But now it's not going to be a cytotoxic cell that it'll be presented to. Now we're going to present this to a T helper cell.